Today we are going to discuss about an interesting milk fermented product known by the name cheese. In this first episode, we are going to cover the general concept which deals with what exactly is cheese, how can cheese be produced, what are the main ingredients present, and how exactly we can classify the cheese. Now, what exactly is cheese? It is a consolidated curd of milk proteins, that is casein and the milk fat, all entrapped into a network. Three common ingredients are used in the production of cheese. Firstly, the milk. Second, the starter culture bacteria. And thirdly, the enzyme preparation known as rennet. These three gives rise to what we deliciously enjoy by the name of cheese. Let's go back to history. Now, probably man would have domesticated animals and they would have known such products like cheese or other fermented dairy products like yogurt. But exactly when cheese came into being or how exactly cheese was known to common man? The story goes that it was the 13th of March 479 when an Arabian man traveling by the desert with his camel was taking along with him milk in a sheep bladder or pouch. Now the whole day long journey with the sun heat as well as the stomach lining which probably contained rennin-like enzymes and the lactic acid bacteria normally present in the milk changed the milk into curds and whey. Now, surprisingly, when the man opened the sheep bladder to consume the milk, he was surprised to find a curd and whey. Now, this curd actually served as a delicious food for him, which he called as cheese, and the whey was helping him to quench his thirst. So probably that was the first incidence when cheese was known to man and came to be into the forefront. Defining cheese would be a consolidated curd of milk solid, all entrapping the milk fat into it in a huge network. Now, knowing briefly about the ingredients of cheese, we would try to classify cheese. Classification of cheese is rather difficult because the milk can vary, not only cow milk, but sheep, goat, buffalo and other milk-giving animals can give the milk and we can make cheese from those varieties. The starter organisms may vary and the processing technology or methodology may also vary. This gives rise to an immense diversity in cheese and that is why proper classification is difficult. But a proper systematic way of classifying cheese based on its moisture content have been devised and we can look into this slide to find out that there are 4 different categories of the cheese varieties based on their moisture content. Further classification is also possible depending on what kind of milk is used as well as whether they are ripened or unripened. Now, let us look into some of these to find out how or what exactly the classification has been done to the cheese. The first category includes the soft cheeses, where we can see the moisture is around 50 to 80%. The cheese percentage moisture here is very high and thus the shelf life of these varieties of cheese are very less. They can further be divided into a ripened variety and unripened variety. Now, ripening takes a long time, so the unripened varieties are very soft and typical example would include cottage cheese and mozzarella cheese. While a ripened variety, an example would be camembert cheese. Moving on to the next category is of the semi-soft cheeses, where the moisture percentage is maintained at around 39 to 50%. These again, like the previous one, can be classified as ripened by mold or ripened by bacteria. Mold ripened cheeses are examples like Roquefort, Stilton, while bacteria ripened cheeses include Limburger and Brie cheeses. The third category includes very hard cheeses and hard cheeses coming to the exact third one is a hard variety where the moisture percentage is maintained less than 39 and in between 35. While the very hard cheese have moisture content lesser than 35%. In the hard category, we have the common cheese variety world over known, the cheddar cheese variety. Now, the classification of these hard cheeses have been done by the way whether they form eyes or not. What exactly is eyes in cheese? Now, after a typical fermentation is over and the cheese gets produced, if further fermentations are allowed, then in the already formed cheese, there will be entrapment of gases which results in eye hole structures. A typical example would be the emmental cheese which has eyes, while a typical example for cheese which do not have eyes include the cheddar variety, the commonest variety. Lastly, with the very hard cheese group, we have the examples like Parmesan and Romano. With this, we cover a classification system or scheme for cheeses. And after that, we are moving on to discuss about the three different ingredients in slight detail. The first ingredient that we spoke of is of milk. Most commonly used is cow milk variety. In the traditional cases, they were using raw milk without any pasteurization or any heat treatment. But nowadays, a heat treatment or a pasteurization treatment is often done and milk quality is checked before any cheese is produced. Milk should be of very good microbiological quality where antibiotics, sanitizers or other such inhibitory substances should not be present. This will improve the activity of starter bacteria. Now moving to understand what exactly is a starter bacteria or a starter culture used in cheese production. Any bacteria or a mixture of several bacteria when incorporated into the milk to bring about certain desired change in the milk community gives rise to what we discuss as the starter culture. Here in the slide we can see the person adding the starter culture into milk. Two such organisms have been also pictorified here, the Lactococcus lactis on this side, while we have Lactobacillus delbrueckii on this side. Other starter culture bacteria for cheese manufacture includes Lactobacillus helveticus, Lactobacillus casei, Lactobacillus lactis, Streptococcus thermophilus, and several Leuconostoc species. Now, certain roles of the starter culture bacteria are very important, and the first among them is the fermentation brought about by the starter culture bacteria. What happens actually is the milk sugar lactose is converted to glucose and galactose first. Then this galactose is isomerized to glucose back, which enters the glycolytic pathway to give rise to pyruvate. Pyruvate by this mechanism gets converted to lactate. This is a typical fermentation pathway and generates lactate or lactic acid in milk. Now, the production of lactic acid in milk eventually reduces the pH of milk, which destabilizes the casein micelles, and production of curd or coagulation is the result. Let us see several roles of the starter culture bacteria in cheese making. First, obviously, we have already discussed is the fermentation of milk sugar lactose to lactic acid. Second comes the role of the starter in cheese ripening. Cheese ripening is usually done after cheese is produced, and we will be briefly discussing ripening at the end. Lowering of the pH, which because of the production of lactic acid in milk results in extended shelf life, safety and stability of the cheese, because if the pH is low, many bacteria will not be able to grow on the cheese and help preserve the cheese. Next, the production of lactic acid also gives a sharp, fresh flavor to the cheese. Because of lowering of pH, the casein micelle is destabilized, and this further facilitates the function of rennin, which we are about to add later. Acid production will also help in more whey expansion and shrinkage of the curd. And finally, to wind up the role of starters, we also see that the starters do not allow undesirable microorganisms to grow in the formed cheese. So with all these obvious roles, let us move on to understand what exactly is rennet or coagulum, which is added in the milk to produce cheese. The first obviously is the starter, which lowers the pH, followed by the proteolytic cleavage of the milk protein casein brought about by the enzyme rennin or chymosin. And this is where we discuss rennetting. The term includes the application of the rennin or chymosin enzyme into milk after the starter has been introduced and kept for half an hour. Now, we are typically looking here into the action of the principal ingredient of rennet, that is rennin or chymosin. What exactly it does? Chymosin or rennin specifically recognizes 98 part or 98th position of histidine and 111 position of lysine in the amino acid chain, building up the casein molecule, kappa casein molecule. And its action is the cleavage in the 105 phenylalanine residue and 106 methionine residue, where the cleavage brings about the disruption of the kappa casein into two components, the para kappa casein and a macropeptide. This macropeptide gets released into the whey, while the para-kappa casein, which is hydrophobic in nature, gets entangled with other such hydrophobic para-kappa casein, which has been cleaved by the chymosin enzyme. Now, this is how exactly the para-kappa casein all comes and aggregates together to give rise to the typical curd, while the macropeptide is released into the whey. And this is where we get the distinction between curd and whey, which we all know during the production of cheese is very, very important. We can see here the curds and the whey separating after the action of rennin has taken place. Now, once we have seen the action of rennin, we are now looking into the sources of rennin. From where do we get rennin? Initially, it was the suckling calves, very young calves, suckling calves. They were slaughtered for the fourth stomach or abomasum, which contained rennin. But the worldwide shortage of such calf, as well as due to ethical reasons, the calf rennin was discontinued, and there were several other types of rennin incorporated, like blends of rennin and pepsin, or it can be rennet extracts from mature cows. These two obviously do not give such results as the young calf could give. Then later came microbial or fungal rennin. Typical fungi like Mucor Mucor pusillus, Endothea species, these produce rennin-like enzymes, and those could be used. But again, to say, these also did not give good result. And finally, we now rest on the recombinant rennin for the worldwide cheese production. How exactly is recombinant rennin produced? Now, a copy of the calf cells are taken from obviously calf, and then the gene analyzed. The gene responsible for producing chymosin is integrated, or rather cut off, and used to integrate into a plasmid. Plasmid is the vector used for cloning, and this recombinant plasmid is inserted into a yeast cell, or it can also be inserted to E. coli cells. Then these are overproduced. The yeast cell or the E. coli cell will start proliferating in numbers and produce huge amounts of this recombinant chymosin, which we can obtain in the form of bottles or as well as in the form of tablets. So far we have seen the starter culture and what they are used for, and heard about what rennet would do in production of cheese. Now, understanding all this, we will now move on to see how exactly a typical cheese can be manufactured. I have a pictorial representation which will follow us and see how exactly cheese is produced in an industry or a food plant. First, we have used raw milk here. Now, in traditional cheese making processes, they always used raw milk and never pasteurized or heat treated milk. But for safety's sake, as well as product equilibrium, as well as homogeneity, we use pasteurized milk. So, raw milk is first pasteurized in today's process. Then, it is allowed for a standardization and a filtration step. The pasteurization step is brought about by the use of plate heat exchangers, which incorporates heating the milk for a specific time period and at a specific temperature. Then it is cooled down and sent for standardization and filtration. In a filtered mode, clarification is also achieved, which improves the viscosity of milk. Standardization is often essential because the milk may not meet the exact criteria for the milk ingredients, which may be increased, like milk fat can be increased, milk casein content can be increased. So a standardized method is used here, which gives the milk proper homogeneity. Then it is pumped into a vat known as a cheese vat, after which the fermenting organism or the starter culture is inoculated into the milk, kept there for approximately half an hour, during which the starter bacteria, as have been already mentioned, will start producing lactic acid. They will begin producing the lactic acid only slowly, and before the curd could be completely formed, we are allowing the addition of rennet. The process is known as rennetting. We have already discussed what rennet would do. Once we are adding a rennet, we will leave on the preparation undisturbed for approximately again half an hour, during which time slowly we will see the separation of curds and whey in milk. After that, the curds are all brought into one side or in one place so that more of whey can be expelled. As well as the curd is then cut into several sections, maybe diagonally, vertically, so that more and more whey can be expelled. Stirring also aids in more expulsion of whey, which will result in compact production of cheese. Let us see the next diagram here. That is, after the compactness or cutting and stirring effect is done, the whole thing is actually seen to lie at the bottom of the cheese vat. Then the drain is opened while the whole of the whey is drained out. We do not want the whey in any of our cheese production, and simply we do with the curd. And it is interesting to note that if you use 100 ml of milk for cheese production, you will just have 10% of it being converted to cheese, while 90% is removed in the form of whey. So we have very less production here, but this is what is desirable. Once the curd has been drained completely of whey, we take it into the miller machine, which mills it into smaller particles so that homogeneity is again maintained. These are then forced onto the conveyor belts, which moves on the slides, and the salt is sprinkled all over. Salting is an important step in any cheese making. In many cheese processes, certain salting is done at a later instance or sometimes can be added even before. Now, this is a typical process where we see just after the milling, salting is done. Salt adds flavor and taste to cheese, as well as inhibits certain molds and yeasts which may have encountered during the production of cheese from the various equipments of the cheese production. If we are willing to use fresh cheese like cottage cheese or cream cheese, we do not allow this to be gone for ripening or pressing. We simply take it out at this level and enjoy fresh cheese. While for most other cheeses, we keep it on certain molds which are again taken on conveyor belts. Now you can see here a cheese press. What it does is, into each hoops, the cheese, that curd is contained, and it is completely pressed. So release of more whey results, as you can see here, and that excess whey is drained off to give harder texture to cheese. we have already discussed that cheese differs depending on the moisture content. So high moisture containing cheese would not be pressed, while low moisture containing cheese definitely needs to be pressed further so that they become more and more hard. So these are then transferred to the ripening area and then coated at times before ripening, after which ripening can take as long as a year or even more. You can see here a ripening area or a zone. The cheese are coated and kept here for ripening. What is this ripening now? Now we have already seen that starter bacteria are incorporated and they have residual effect. Now, if you keep cheese after production for a week or two, these starter bacteria will not live anymore. They tend to die after a week or two. But during the first or the second week, their proteolytic and lipolytic enzymes and the consecutive activities of the proteolytic and lipolytic enzymes enhances the flavor, texture and aroma of cheese varieties. So ripening is often desired in most cheese production units. To conclude, we have a platter of cheese for you here. You can choose among the different varieties that suits you the best. But remember that during production we have seen what are the common ingredients that make up the cheese, the several classification varieties of cheeses and what they are mainly meant for. They are actually high in milk fat and high in milk protein. So be careful when you consume them and wait for us in the next episode, which will cover some more of other varieties of cheeses.